Okay, hello everyone. This is Rafi, and I'm here with Gabor. This is somewhat of a Break Nutrition podcast reunion in the times of COVID to just shoot the shit and talk anything that comes to mind with regards to COVID and science and hopefully immunometabolism. Maybe that's going to be our theme, or maybe it's going to be angry political ramblings. I'm not sure yet. Maybe we can do both. Let's see where the night takes us. So thanks for taking the time, Gabor.
Sure, no problem. I think if you want to take it to the political direction, then it's most likely I'm going to be a listener, it's not really a discussion because I'm not much into politics. It's difficult to avoid these days, but I think it's kind of the clash of science and politics what's been happening over the last couple of months or almost a year now. And many politicians pose as following the science, and nothing is further from the truth. And some scientists grab the opportunity to come out to the limelight and pose as with probably political aspirations for later. I don't know, but they really like the spotlight and like to give these interviews to mainstream media and often talk a little bit of bullshit. But yeah, there are also vested interests. Some of these scientists are known to be involved in antibody testing, antibody treatments, PCR testing, or vaccine developments and various attempts, which can easily be considered as conflicts of interest, I think.
Yeah, I think the conflicts of interest abound. I think, of course, the politicians, as expected, are going to follow the very obvious incentives that they pretend aren't ruling them and their actions. But, that's not really where my mind goes to first. Actually, I'm much more disappointed with the general scientific establishment as one. I think that's maybe where it's more jarring to me to see a lot of doctors, a lot of nurses lose contact with reality when comparing year on year how their service was affected, and maybe being totally unable to, get outside of their anecdotes, however telling it may be, and actually, fulfill the role that they're doing in public health, which is being informed. First of all, I think that's everyone's responsibility, whether you're a nurse, you're a doctor, or you're a researcher. I think your basic level of information preparedness during a pandemic remains a primary responsibility. And, just help to drive the fear and not, even if you don't know, you don't need to have the answers, but what you do need to encourage is the proper approach, the proper consideration of the science. And I think that's the only thing that I would have liked to have seen. I didn't need nurses and doctors to be qualified epidemiologists or immunologists. That's not what I was expecting. I just would have liked the defense of the basic scientific principles that go into assessing this. So that was where it was more disappointing for me personally. I don't know if really the scientists let us down first. Who knows? Because we don't even know the origin of this virus yet. It's still in dispute whether it has a lab origin or whether it has
I have my suspicions, but let's avoid
Oh, let's go there. Let's go there.
theories. Let's go
I
there.
want to go there
Okay.
because it's, the problem is I don't think it's at the stage where it's ripe for conspiracy. Because I think the level you and I are looking at it is where were the cells passaged in, from what species and that sort of stuff.
I had followed mainstream opinion for quite a long time. I think it started in March when you saw the papers appearing in Nature Medicine and other high-profile papers or journals that it cannot be lab origin. These are the proofs.
I was dismissive. It was based on some sequencing that I hadn't looked into, but I was dismissive.
Based on several things. But at that time, I was not really into the background science, so I couldn't even judge properly if they were right or not. And then my focus had been on other aspects of the epidemic, and the usual metabolic problems and these kind of things, and looked into masking and
Right.
other weird things. A lot of influenza stuff I had to read back on, because I just hadn't been following this part of science up until Q1 this year. So, and then suddenly sometime during the summer, during some, when I do some research, I read a lot and I check the references, I check the citing literature, and I go about for hours on. Sometimes I can spend hours on one paper, reading the references, reading the citing literature, clicking on links,
Oh,
related
you
articles, and
can. I'm sure
whatever.
you can spend a day or more. I know it's
And
happened to me
yeah, yeah, yeah.
where you
And
spend more than
and
a day on it.
suddenly I found a paper which was looking at peptide mimicry between the spike protein of SARS-CoV-2 and different potential host animals. And it didn't seem interesting at all. For the first reading the abstract was okay, but then I started scrolling through, and when I got to figure 1, something was just striking poking out from my iPad screen, and it was two columns with very high level of mimicry, and it was based on different hosts, and the mimicry was very low. It was totally not similar to other coronaviruses. It was not similar to non-human primates. That was not similar to pets like cats and dogs and whatever. And there was, I would say, extreme mimicry between humans and the spike protein, which is definitely not explained by the time it's spent. I mean, confirmed time spent in humans, like from last year,
Yeah,
October,
basically a
November
year
last
ago,
year.
yeah, or a bit more.
Yeah, the paper is from the summer, so then it was
Right.
even shorter time. I think they submitted it during the late spring, so it was half a year. And that level of mimicry is implausible in half a year. And then what was even more striking, it was exactly the same level of mimicry with our good old friend called musculus, which is the common house mouse,
Right.
which is rather interesting, because just a week before that I had read another paper which discussed that okay, we were not able to use regular mice for lab experiments because regular mice had so different ACE2 receptors that mice were not susceptible to SARS-CoV-2 infections. So they had to develop actually humanized ACE2 mice, but that happened actually back in 2013 or 15 or so many years ago, and for SARS-CoV, the first one, which was spreading in 2003.
Yeah.
So after that, they developed these humanized ACE2 mice so that they can use them in the lab. So my first thought was, how was this extreme level of mimicry plausible between mouse proteins and viral spike protein? Because the mice are not a host, a known host for this virus. So nobody implicates mice as a intermediate host, for example. It's implausible. And then I think only one plausible explanation remains: these ACE2 humanized mice. But these mice doesn't exist outside of a lab. So
Right.
what's going on then? But I
Yeah.
mean, that level of mimicry suggests that the virus spent a considerable amount of time, or I mean, considerable cycles of replication, number of cycles of replication in that kind of host, or at least cells, because otherwise it won't develop. You know, this kind of mimicry develops when the virus is budding from the host cell, and it is released with the same it's very similar glycosylation, and over time even the genome adjusts to the some of the host proteins, or at least some of the host peptides, some parts of the host proteins. So that was a kind of a shock, and that was the time when I started looking for information, if there is anything else about a potential lab origin. And then, of course, if you start looking, you start finding, and there are some other boggling coincidences and piece of information that points to that direction. So I wouldn't dismiss lab origin at this stage. I'm not saying that this virus is certainly of lab origin, but I don't think that many of the initial explanations, retrospectively, these more seem like explaining away some odd findings than plausible explanations. And
Right.
you are not really welcome to discuss this openly, although I think everything should be discussed openly in science. So there are some exceptions. You can follow Alina Chen, for example, on Twitter. She's in a MIT lab, if I'm not mistaken, with some colleagues. They are doing some investigations, and there are also some whistleblowers from China telling some not implausible things, because I think that the protein, the peptide mimicry points to long time, which is most likely could come from passaging, or whatever it's called in I think it's called the same way in English.
Yeah,
So
where
passaging.
yeah, which is if people don't know, it's where you basically you have the cells in a little petri dish and you introduce the virus to them, and the cells basically go through replicative cycles with the virus, and they produce the virus, and basically you get that interaction, the passages.
Yeah, yes and no. I mean, if you look at the whole virus and how it's different from any potential long time predecessors, it definitely was. It was definitely shaped by a working immune system. So it's not only cell culture. It should have been. It should have spent time in humans, based on the peptide mimicry at least,
Right.
and in mice. And in mice, of course, mice are not susceptible hosts, so only ACE2 humanized mice come into the picture,
Yeah.
and that is a strong one for lab origin, or if it was not engineered, but it spent some time in a lab accommodating different organisms,
Right.
and then probably escaped or whatever, I don't know. But there are some more details. For example, the furin cleavage site. The chance to develop a furin cleavage site via passaging, I think, is extremely low. So that's another suspicious point, change compared to previous. I'm not aware of any other coronavirus which has the furin cleavage site.
Okay.
We know, for example, HIV and other viruses have this, which increases their virulence greatly. and this virus somehow just obtained this furin cleavage sign between those two spike subunits S1 and S2, which is, I would say, rather intriguing.
Right. It's from what I had understood initially was that the sequencing had been done and there was no sign of CRISPR manipulation, essentially. However, I think that's actually that's not necessarily the only way that you can introduce mutations. Like we said, you can do it by having it evolve in another organism, and then the marks that you're looking for aren't necessarily going to be of the same nature as a CRISPR clue would be when you're sequencing the genome. So I think that introduced more doubt to me and opened up the possibility that indeed it could have been evolving in other hosts, and the ACE2, the humanized ACE2 receptor in the mice could be that missing link to explain how they could have in fact done that. So that's really interesting, I think.
Yeah, so that's where I am regarding at least the origin. And you know, most likely everybody read about the Yunnan, or whatever it's pronounced, the Chinese name. It's a
Wuhan?
prefecture in China. No, the Yunnan.
Oh, the Yunnan province. Okay.
Yes, where
Gotcha.
the mines and the caves and the beds are, which is more than 1,000 miles away from Wuhan, which in itself I think is a little bit intriguing. So when you have
Right.
all the viruses in southwest China, and then you have you have an outbreak in northeast China, then it's rather intriguing. But
It's just fascinating how close the market and that lab were, or are
yeah,
actually. It's
yeah,
just
yeah. But now I think nobody believes that the market was the actual starting point of the first infection occurred somewhere close to the market, but not at the market.
right. I think you'd
So
have to have a very specific evidence for that because it could have so
and
much more time elsewhere to evolve, basically.
yeah, and also if you know that Harvard study which looked into the hospital movements satellite data in Wuhan, and this unusual surge in hospital visits started end of August last year, and then it
End
went
of August. Oh
through
wow.
it went through all September and October. So this purported December outbreak may not be, or there were two viruses or whatever, but something strange also happened a couple of months earlier.
And in Italy, it appeared in
According to,
November. They have from blood samples now established it appeared in November.
yeah, new study claims that it must have been there since summer last year, because they measured antibodies starting from August. And then September, and then the interesting thing is that antibodies went down, and then it started. The resurgence started in January only. So yeah, it might have been two viruses, and then it could explain a lot of pre-existing immunity, especially in Asia, where
Yeah,
spread
that's
is
true.
expected more widely. But this is pure speculation at this point. There is a lot of attack on this paper, which claims that in Italy the seroprevalence was high already in September last year. I think I don't know. Their methods seemed okay.
You know, the thing is, we have there's so much immunity in the young that we're not going to detect by looking at the IgM. That it's, you know, it's I'm not so sure yet that we're very good at it. We can do some pretty decent estimates based on testing a population with an antibody test. That, if we're talking about immunity at that level, we can give a pretty decent estimate, I would say. But to estimate the real one, taking into account the people who are going to have an IgA response, you know, in the mucosa of their throat and lungs and nasal passages and not have it show up in the blood because they've neutralized the virus that way. That's probably not a minor part of the population. So it's hard
So
to
far, it
estimate.
seems, at least it seems that even asymptomatics develop antibodies.
Yeah,
I mean, systemic
right.
antibodies.
Yeah, yeah.
Most confirmed asymptomatics who were later measured for antibodies were positive, the vast majority. So those claims that talk about people just repelling the virus without any measurable. I mean, of course, you could measure in the mucosa, but it seems that eventually they also develop some systemic antibodies.
Yeah, I
So
wonder about that. That would be nice to know more precisely. But you know, the thing is, we rarely confirm those population samples with like viral cultures, which would be the gold standard. It's rare to be done, but when it's done, it's really telling. And I was just reading Carl Hennigan's review. You know, he submitted. When you look at what's being done with viral cultures, you see that the numbers are quite different from what you obtain in the field. So I think it's really something we have to start to do more rigorously, because you end up wasting so much time repeating large scale tests without ever improving your certainty in your results, basically. So that
yeah,
in
but
terms of
viral
preparedness,
cultures can be done only in BSL 3 or 4 labs.
right,
So
right,
it's not so easy. And then you know, there's a difference between, at least as far as I know, there's a difference between different cell cultures you use. So if you use viral cells, for example, they are much more susceptible
right,
to infection.
yeah, correct.
So they will show up much lower viral titers than human epithelial cells, for example. So
But that's okay for a test, because all you need it is to be consistent. So when you're looking for PCR, for example, as long as you're using the same kind of cell line and you know what kind of level basically you need in order to obtain sufficient live viral culture to produce a culture, then you can standardize it. So you're right, from a model perspective, you want it to infect human cells, not vero cells, and that's definitely important. But to standardize a test in terms of precision, all you need is to actually standardize it. Then you can choose different cell lines, I think.
yeah.
So it depends what you're trying to answer, basically. But from a mass testing perspective, like to establish an evidence base just of reliability, I think we like it's crazy. There's this very long article by Yeadon, you know, the guy from Pfizer,
Yeah.
explaining a lot of the lab procedures that's going wrong in these places in the UK that they've set up on a very short schedule. It's a mess. It's a mess. You just cannot trust what's coming out of there. It's amazing.
Yeah, it's
Yeah,
there are a lot of different opinions, and when it comes to researchers with very little public health or broader problem-solving experience, you see that they just maintain this narrow research lab perspective. I just read
yeah,
the other day kind of a scientific opinion piece by a Australian virologist who said that look, our lab doesn't see meaningful levels of false positives. All the other labs in Australia don't see meaningful levels of false positives, so there are no false positives. I
right.
think this is not really a practical way of thinking,
Yeah,
because Australia, especially without the levels of mass testing going on in other countries, in other larger countries, it runs only a limited number of tests, and they have, as far as I know, they have single protocol supervised by a scientific body, and because they haven't been overwhelmed with these tests. But in many countries, to increase testing, they made huge compromises.
Yeah.
They involved labs which do not really stand up to scrutiny.
They didn't hire qualified scientists.
Yeah, and they sometimes I saw in Italy when these surges occur in infections, they just let go some of the genes which they are looking for in
Yeah,
the
exactly.
PCR.
Just
Without a
so,
control,
right.
without triple genes being followed, they just look only for one gene. Then your sensitivity decreases, and the specificity also decreases. So your false positives go up like crazy.
Right.
I
So
mean,
let's name
like,
it for people. You have the membrane protein, the nucleocapsid, uh, uh, um, uh, uh, parts. Then you have the membrane protein, the spike protein. So you have some options, and now we have some tests that are going down to just looking at one, like just the spike protein, for example. Of course, you're going to get a lot of nonsense results from that. You have to, and there's a reason why we were doing, I think, triplicate, right? We were doing 3 different genes, basically.
Yeah, and the proper control.
And proper control.
Yeah, and I think so. Yeah, technical false positives can be a problem in some countries, in some labs, and definitely it has been a problem in some countries and some labs. It's not a blanket
Yeah.
observation that
like the NHS was doing fine
we also
initially.
know about we also know about labs which run at an extremely low overall positivity level. So if their positivity is 0.2%, it's quite obvious that your false positive rate cannot be 0.8 or 1%,
Right.
because your overall positive rate is 0.2.
Right.
So these labs are running just fine, and technical false positives is not an issue. But it's, I mean, there is, I think Kevin McKernan calls it epidemiological false positive. Right. When they run it to very high cycle threshold levels, and they call everybody a case who has a positive PCR test result, and many of these guys are almost certainly not infectious. So in an epidemic, we should focus on infectious people and quarantine infectious people, because when you start quarantining basically everybody who presents with a positive test result, and up to 90% can be non-infectious in some circumstances, and then you quarantine even their other household, all their contacts. Right. Sometimes all the close contacts are quarantined as well. So you quarantine masses of non-infectious people. That's certainly worrisome. I wouldn't be happy if I was quarantined based on a friend's positive test, knowing that the friend was over the infection weeks before.
Yeah, there are PCR tests that will catch it up to 78 days after the first onset of symptoms. So really, you can sort of make magic from it. It's this very powerful amplificator, and its power is also its downside, which is if you set those cycles too high, around 30, 35, even 40, you're going to completely overshoot, because actually most of the true positive and true negative samples you're going to get, basically the estimate I've come down to is around 23 cycles seems to be a very reasonable place to set it. And it can be adjusted, right? You can have internal standards in the lab and decide to adjust it, but around that level, certainly not 35.
I think very rarely you can find infectious, so-called live virus at a cycle threshold of 35. But this occurs mainly due to the fact that some pre-symptomatics are also measured, and
Yeah.
they start shedding live virus when you measure them. And of course, it's important to capture these pre-symptomatic people. So I think a good compromise could be made that you use a cycle threshold of 35, but if anything between 25 and 35 or whatever threshold, I think it's lab dependent to some extent. You can draw a line in the sand for any lab that between 25 and 35 you should repeat the test 48 hours later, because then
Right.
you will see 48 hours later you will see that the guy has more virus or more viral RNA or
Right.
less viral RNA. So
Exactly.
what is
He's
the actual
set the baseline.
trajectory?
Yeah.
Yeah, what is the actual trajectory? If
Right.
this guy is getting out of disease, or he has just started shedding even more viruses, and then he has to be or she has to be quarantined. That's fine. But without doing so, it creates a huge mess. And also, I don't know why it had to be thrown out the window, but positive test results had never been called cases
Right.
before 2020,
Yeah.
because
Let's
positive
explain
test
that,
result.
because it's a complete total inversion of what's been done before. So what's a case? Like, let's define a case.
Normally, a case is somebody who presents with the symptoms of a disease.
Symptom
And
based.
then yes, you have the typical symptoms, and then you are verified with some lab protocol that you seem to have the infectious agent. You are virus or bacteria positive according to some lab protocol. Then you are a confirmed case. So you are a case if you have symptoms, if you present with typical symptoms in the middle of an epidemic. So if you know that we have a SARS-CoV-2 epidemic going on, and the guy presents with a fever, dry cough, muscle pain,
Headache.
diarrhea, or whatever typical symptoms, then he's a case. And then there is a test done on the folk, and if it's positive, he's a confirmed case. So I think that it has been as simple as that for decades. And suddenly everybody with a positive test is called a case.
Case. Yeah,
Case of
exactly.
what?
Completely
Uh,
forget Cox postulates, not paying attention to anything we would do with influenza or any other infectious disease, and just
it's a
completely invent the rules.
case of showing viral particles. That's the exact definition.
That's the crime.
Uh, Yeah, does it make any sense? Who is
Yes, if
a
you
case?
want
Who wants
to
to be a case of showing viral particles?
if you want to drive fear, it's genius. Show people that you're doing a lot by doing a lot of testing, yet ensure that you have the excuse to keep doing what you're doing to,
Yeah. So
exert
in
control.
fact, it's fascinating that we usually condemn lower sensitivity methods for identifying whatever markers or pathogens. But in this case, the quick tests make a little bit more sense,
Right.
because
Lateral flow.
for the price of a single PCR, you can do, I don't know exactly, 4 or 5 quick tests.
Yeah.
And doing a quick test for 5 days every day for a single PCR, you can capture a lot more useful data, and you omit the useless data. Because the quick test device has lower sensitivity, and this way it won't be positive when you have very low viral loads, or you have no viruses, just viral particles, of course. Then it won't show up as positive, or it will show up as positive much less frequently. And actually, using 4 or 5 quick tests seems to make a lot more sense now, based on our experience this year, than doing one single PCR, especially when omitting genes and the control runs and the control genes and these kind of things to be able to run more tests.
Yeah, and it's really a pity because there's nothing inherently wrong with PCR nor the lateral flow. It's just a question of using them at the right time. And this was one of the points that Dr. Claire Craig had made about using the PCR as it was at the start of the epidemic, where it's very sensitive. That's okay because you really want to find every positive case and quarantine the person who's positive and is coughing, right? That makes sense. You're okay with the bias being in that direction. However, the later part of the epidemic, you have to adjust your cycles maybe a bit more rigorously and lower that and understand that the sensitivity is going to be not in your favor now that you've had a first wave of a high increase in immunity and thus a lower amount of susceptible people in a sense, right? And a lot of people who have passed away, unfortunately, and those sort of things. And then, like you said, even if you combine it with the lateral flow, for example, doing PCR maybe at around 25 cycles or just around there, and then confirm with the lateral flow, which is cheap and you can repeat. Do some confirmatory testing. Don't base it without the symptoms. I mean, there's a lot of basic stuff that can be done to avoid, I think, the pseudo epidemic, but maybe address the ripple, as Yeadon called it, right? This ripple, which is in fact a resurgence that actually we should talk about, because let's talk about masks and let's talk about seasonality of respiratory viruses, because I think there's a lot there that is going to be important in order to understand what we're going through now and why it was predicted, and maybe the masks had this effect. Again, unintended consequences. We see this all the time. And we were talking about this before recording, so why don't you go with what you were telling me, Gabor?
About seasonality? Or?
Yeah, and especially about the places that lock down harder, masked up harder, and are seeing some resurgences.
I live in a country which is a prime example with some of the neighbors which locked down early and very strictly. So, for example, comparing to the UK, Hungary and the Czech Republic locked down a week earlier, and while the viral spread was I think behind at least 2 or 3 weeks. So that's almost a month overall, if you can imagine, because these are smaller, a little economically less advanced, less travelers, less busy airports, and so on and so forth. So these countries were behind in the initial spread by several weeks.
Mm-hmm.
And then they locked down a week earlier and very strictly, and then people were scared, and that adherence was really high in these countries.
You could argue that lockdown was more likely to work in that case, where you had some foresight before there were some positive cases.
Yeah. I think we just followed other neighbors. So these countries are always look at neighbors to the west. Germany, Austria, and what they are doing. And while the spread was behind by several weeks, they made the same interventions at the same time, basically, or only one or 2 days later. So I think actually I'm of course no fan of lockdowns, because these do more harm than benefit. These just delay the problem, especially if you are early into the spread. Then okay, but that's another topic of lockdowns. But theoretically, if people start moving and everybody's at home, it should work. So I'm not a denier that a hard lockdown could slow the spread of the virus. And it seems
that it didn't stop prisoners from being infected, even when they anticipated it.
Well, it depends on when the lockdown is launched, because now we know, for example, that in countries which have more tentacles to other parts of the world and more travelers and more busy airports and so on, these saw the surge of the infections much earlier, and they locked down late. So if you lock down late and you have the disease in an advanced state already, or not the disease, but the pandemic, the viral spread in an advanced state, you can't prevent it anymore.
I'm
That's
starting
quite
to
obvious.
think you want to be Hawaii and the infection starts in Wuhan and you're notified a week in advance, and then maybe lockdowns could work.
Yeah, when
I'm
you get
prepared to believe
when you
that.
notice, yeah, when you get the news about the first out of China transmissions,
Yeah.
you lock down. If you're an island, Hawaii is not a good example because you have a lot of
Yeah.
income from tourism. Basically, I don't know how many percentages wise,
Just
but
close everything down. No planes in or out,
that's
just food.
yeah. You can do that, but if it's questionable if it makes sense. Still,
Yeah.
I don't think so. But
I'm doing the
I think
devil's argument for lockdown.
purely theoretically, it could work, and it seems
Yeah.
that it worked. It did work in some countries, and but the problem is that the price of the lockdown is so high,
Right.
the damage is so big that when you realize it afterwards, you decide that never again. So these countries, Czech Republic, Hungary, decided, okay, we cannot do another lockdown. So what should we do? We will do a partial lockdown or masks or whatever. We try to slow the surge, but no lockdown. And then these countries seem to experience a even higher surge and deaths and everything than compared to, for example, Sweden. The Czech Republic surpassed Sweden on the mortality charts a couple of weeks ago already, and Hungary is about to surpass Sweden in a week or two. I think 2 weeks, in 2 weeks' time. So it seems that there is no way out.
Yeah.
If you are in the middle of Europe, if you still want to do some exchanges with other people, partial border closures are just stupid. You won't stop the
Yeah.
virus if you do partial border closures. You have to stop all travel so that the virus cannot come in. If you
And
manage to.
but even internal travel, doesn't correlate with any,
Yeah, but
any changes.
during the summer, the virus almost disappeared.
You're right.
Whether it truly disappeared is questionable because
Yeah.
there are some hypotheses that the virus actually lays dormant in some nerves or immune cells and just reactivates when conditions are more favorable, and then we are
Sure.
back to seasonality because it's a funny thing. Most people imagine that seasonality depends on weather. And
Yeah.
then this chain reaction somehow slows down during the summer, and then the chain reaction of transmission accelerates during late autumn and during the winter. But quite some evidence points to linear transmission of these viruses. So it seems that the famous examples when in the 60s there was a staff shift on a Antarctic research base. So the complete staff was replaced by new staff. So everybody left the place before winter came, and then a new staff was placed in the research base. And after 17 weeks, a coronavirus outbreak occurred. So how do you explain this with linear transmission? After
Yeah.
17 weeks of complete
17.
isolation,
Wow.
that's 4 months.
And they were living in a fridge, basically.
Yeah, they're living in minus whatever
Yeah.
degrees, completely separated from the whole world in Antarctic winter.
Amazing. This,
So
you know,
of
this is
course
why
they were.
I always come back to what I think it was Osterholm who said you know, trying to stop it, it's trying to stop a virus is like trying to stop the wind. It's not something you can do really fundamentally. You can
Yeah,
protect
yeah, but
yourself from it, you can barricade, but you can't stop the wind. The wind is gonna
the
go through.
examples of these countries, and of course we have New Zealand and Australia and some other countries provoke to speculate about pre-existing immunity, but there are some countries where extreme isolation seems to work, or we have some evidence that it probably worked. And then you start thinking, we see these examples, and then we see these lockdowns appearing to work. And then the idea came to my mind that perhaps spread during a pandemic, when a new so-called novel virus is spreading with substantial susceptibility in the population, the spread indeed more closely resembles a linear transmission, like a chain reaction. And then you have the R number of how many people one infectious man can transmit to, like 2 point something or 3. That's an average, of course. And then you measure these things. But as soon as the virus starts becoming endemic, so establishes itself in a population, it becomes a mix of linear transmission. So I cough and you inhale and you get it, and so much that the traditional view of how viruses replicate or transmit between animals or humans. And then there is the other one where the virus lays dormant or just lays partly inactive or being controlled by the immune system or whatever happens, because we don't really know precisely. For other viruses which have this dormancy or latency, we do know about how it occurs. For example, the herpes viruses, how they lay dormant in the nerve cells, and other viruses in other cells, and then reactivate when the immune system becomes suppressed by another infection, for example. And then you have a herpes or whatever it's called. What is it called
A
in
cold
English?
sore.
Yeah, a sore. Then it reactivates when you have another viral infection, for example.
Yeah.
And I cannot exclude that something similar happens with respiratory viruses. Probably not in nerves, but it cannot be excluded, because it is already described that, for example, from the nose, SARS-CoV-2 goes up to the brain through the nerves. It can go up, I mean, and then that's associated with severe disease. So it could lay dormant in immune cells, for example, or epithelial cells kept in check by cytokines or whatever means. I don't know, by whatever means. We don't really know. But it's very funny when I think that the most recent WHO commissioned review on review of non-pharmaceutical interventions in the general public, which was published online in February, so just before the pandemic. Probably it was finalized late last year. But anyway, it's the latest WHO commissioned review, and it was done by Hong Kong University researchers. So, you know, a masking country, masking city. And their conclusion is extremely interesting. They found that nothing seems to work. Masking in the general population doesn't seem to work. Hand washing doesn't seem to work. And other non-pharmaceutical interventions in the general public don't seem to work. So this is the basic conclusion. And then they start kind of speculating in their last one or two sentences already in the abstract. I don't remember word by word, but they speculate that, do we properly understand the transmission of these viruses? because these should work. It's just common sense. I mean, if you put a shield in front of your face, you cannot transmit a virus. And the conclusion is that we most likely don't properly understand the transmission ways of these viruses. And this is exactly what I started thinking about as well. That there's as soon as there was at least one big wave of infections, the virus kind of establishes itself as endemic in at least part of the population. And during the summer it's suppressed or lays dormant or whatever. But when the conditions change, and these conditions may change the virulence of the virus, as well as the activity or potency of the immune system. So these interactions between virus and immune system change in favor of the virus, obviously. And then when it happens, and then we can go into the details of seasonality, because there is a lot of information out there, mostly in connection with influenza, but also for other respiratory viruses, that how their membrane softens up or hardens with colder temperature, or softens up with warmer temperature, and they need this soft jelly-like structure or texture to merge with host cells, but they need the more rigid version when they spread in hostile air or outside environment. So that's why, for example, why they may favor colder temperatures for quicker spread. But it also has an impact on the immune system. It was shown that monocytes and macrophages, for example, these viral particle gobbling cells, that their motion slows with lower temperatures. And there also humidity. Humidity impacts the stability of the virus in aerosols or particles. But humidity also impacts the flow of your own mucus. Your upward flow. It's a continuous upward flow of mucus in your respiratory system, and then you swallow it, and there you have the connection to the digestive immune system. You continuously swallow your own mucus coming from the respiratory system, and if the humidity is above a certain range, or also is below a certain range, then the flow of the mucus slows down. And if it slows down, it provides a better environment for the virus to bind and attack cells below the mucus. So these are very interesting findings, and there are
You need
also
a
many other
higher
things. For
mucus
example,
throughput.
air pollution, the impact of air
Yes.
pollution, the impact of pollens, for example. Pollens interfere with viral particles, and they reduce infectivity of viruses. So, for example, this may be one of the mechanisms during the summer when you have higher pollen levels in the air, and then this interferes with viruses. So there are many environmental factors which interfere with both the immune system and the virus infectivity. And this basically, we don't fully know, at least I don't fully understand yet, which has higher and which has lower importance. So how this is distributed between different parameters mentioned already, humidity, UV, for example, we forgot about UV, temperature, pollens, whatever, many factors, the importance of which is not fully clear yet, but we do see that these have a substantial impact. And we also spend more time indoors, of course. When weather is colder, we spend more time indoors, when the aerosols facilitate viral spread. So these are pretty well understood, perhaps not the details, but overall well understood.
Yeah, I think for me, what it always says is that there's probably a lot you can do for your own internal terrain, and how inhospitable you can make a life for a virus in your body. Maybe enough just so that you can beat it back. And I think this is the public health message that hasn't gotten enough
No,
emphasis.
absolutely not. I think you can influence these external parameters a lot. It was shown, for example, in connection with influenza, that just airing the room regularly and intensively is worth at least as much as a influenza vaccine.
Right.
So it reduced transmission by 50-60%. Just airing the room. This is only one parameter.
And that's
then
a
you
lot
can,
because you have a 50% chance of transmitting it to a person that you live with coronavirus. So that means you'd be left with an absolute of 25, which is much better.
Airing the room is a huge one. You can do it very easily. Another one is setting a fairly stable humidity level in your apartment or house. It's easy because it should be around 50%. More broadly, 40-60% humidity, relative humidity is ideal. And of course, what happens during winter, during most older houses with traditional heating systems, your humidity gradually drops when you start heating and stop airing your rooms. Stop airing favors the viral aerosols hanging around, but it also favors even the infrequent airing of the room favors dropping the humidity, because you lose absolute humidity, relative humidity. You lose some water in the air every time you let in cold air, because the cold air contains much less humidity.
Right.
So when you heat it up, the relative humidity drops. So this is what I've been experiencing for this heating season. I was carefully watching. We started out around 62%, and now we are down to 37%. So I should already start a humidifier to push it up above the critical 40% relative humidity. And these are relatively easy things. And then the elephant in the room is your own immune system. How you can support your own immune system, of course, because that's where the biggest differences you can see when you talk about susceptibility to this virus. You see the age is the single biggest one, and then you start with the sex. Male sex is much more susceptible, and then the next one is, of course, all the comorbidities, so-called comorbidities, basically metabolic diseases, to put it shortly. So, and I just see people all over the internet whining about how they are scared and they will get the virus. And young people, young ladies, for example, it's crazy. I mean, they belong to the 99.99% of not falling for this virus, and they go around and keep whining about why others don't do something for them.
I think they're scared of being blamed for infecting people more than being scared of
Not really.
actually
They are
dying.
scared about getting the virus. I every day I see somebody on my Facebook wall or wherever crying about oh, well, everybody around me got the virus, so I will be next. What's going to happen? Yeah, and the first question I ask, what have you done? We have March, now it's December. You had 9 months. What have you done to increase your chances of mild course of disease or asymptomatic course of disease? What have you done? What could I have done? This is the normal reply. That you could do a lot. I mean, you can lose some weight, especially if it's around the middle. You can increase your vitamin D. How much time you spent in the sun during the summer? It's too late in early December. You have no chances in the moderate climate, North Hemisphere these days. Absolutely no chance. So there was a lot of time to intervene.
People
To
could do
support your immune system nutritionally, metabolically, with sunshine, with good sleep, reducing the stress. Because if you are stressed out, that will suppress your immune system. That's been long known, that if you have chronic stress, your immune system is being suppressed because it's a corticosteroid. You get corticosteroid when you go to the hospital with a chronic infectious disease.
Yeah.
To suppress the immune system, because after a while it's the same with this virus. After a while, the overreactive immune system is a much bigger problem than the pathogen itself.
Mm-hmm.
So if you have an overreactive immune system, what do you get? You get a corticosteroid.
Yeah.
And what you have, what you show when you are chronically stressed out, high corticosteroid. So it's just common sense, really. Common
Mm-hmm.
sense works most of the time.
Yeah. And you get that Th2 dominance where you're going to overreact, most likely.
Yeah,
And
you
I think that's
see
a problem in.
everything. After a while, the body tries everything to remove something which is purportedly there.
Mm-hmm.
It may not even be there anymore, but this chain reaction just doesn't want to stop.
Yeah.
But this has good reasons. I mean, if you delve into the immunometabolic part, how these immune cells operate normally, how they react to different types of triggers or stimuli, and how they behave metabolically when the inflammation is in the resolving phase, then you start understanding a little bit better what happens when you have high blood glucose, when you have high blood insulin, when you have high blood leptin, and when you have insulin resistance, for example, what role insulin plays in the initial response of T cells to stimulation. And when your T cells are insulin resistant, they don't react properly to this insulin stimulation, and so on and so forth. So there are many details which puts this into a little bit different perspective, and then it starts making sense that why all those people with metabolic diseases are highly susceptible to this infection, and why age, and especially males are not able to recruit or stimulate T cells properly anymore. So the
Yeah.
difference between sexes can also be explained.
And it's also apparently it was biological and not chronological aging that would best predicted the poor outcomes with COVID, which actually I would say further supports the link between the comorbidities that we're seeing, like diabetes and obesity and all of those things that, of course, essentially age you in one way or another. So there's so much there that could be mined for, you know, for lowering the risk. I mean, we could literally change the infection fatality rate, right? If we follow the different guidelines. And
Yeah, well, I think
you
we
know,
have had
we're not doing
enough
it.
time.
Yeah.
Of course, if it started out in March and people were not aware of this, they could do nothing. So they got the virus early and they were susceptible. They couldn't do much about it. But then all the information started to see the light that the obese are much more susceptible. People with diabetes are much more susceptible. Um, and high blood pressure and so on. There are many, basically all metabolic diseases.
Yeah, and this doesn't bode well for vaccines. Vaccines work to the extent that they work because they take advantage of what the body can do. That's what a good vaccine does. It's not, you know, doing everything itself. It creates an initial response, and then the dominoes have to fall after that. And when you have an imbalanced Th1, Th2 system where you have Th2 dominance, and you're going to get hyper responsiveness, and you're not going to get the pro-regulatory aspects of the immune system. You're just going to get a lot of, let's say, discriminate gunfire. You're also not going to be able to start producing antibodies to it. You know, you're going to be you're going to activate the NLRP3 inflammasome, which is actually interesting because one of the things that lower the P3 inflammasome is being in a fat burning state where you produce beta hydroxybutyrate, which can bind to the HDAX basically, and through its methylatory action, actually lower inflammation. So you have to wonder, people who've never seen a ketone in their whole life, of course they have no defense against this sort of inflammation.
Yeah, the good thing is when you have ketones in your blood, you are kind of enforcing oxidative metabolism.
Right.
So even experimentally, it was shown that enforcing oxidative metabolism in T cells greatly reduces the susceptibility of model animals to severe influenza, for example.
Mm-hmm.
So this is absolutely crucial, and this is one place where many people with high insulin, high blood glucose doesn't bode well, because many people don't fare well when they see such a virus, because their initial activation of the immune system will be really sluggish, and they won't be able to recruit enough specific T cells, for example, and they have too high neutrophils already, and macrophages activated or suppressed by high insulin. So their kind of immunometabolic milieu, the internal environment, is already skewed to a certain direction, which doesn't favor a quick and efficient immune response.
Yeah,
This is what we see.
Yeah, go ahead.
This is what we see if you fail to mount an initial strong antiviral response. You are into this long COVID and severe disease trajectory.
So this is really interesting because, one of the things you see when there is a response to a fecal microbial transplant in a ulcerative colitis patient, better outcomes are seen in the people who mount a fever. And in vaccines, it's not uncommon to induce a fever in people. And if I'm not mistaken, it is also the case that with vaccines, you tend to see, a good response if it's sufficiently strong, essentially. And sometimes that comes as symptoms, unfortunately. But that can be a signal of proper response. So, I'm just wondering what that means for how we should be medicating people, because
it's a brand new area, I think, of research, and it's not very popular so far, that the microbiota can have a say in, for example, vaccination. But it's not only vaccination; it's also to natural infections. And I'm sure that you will find a study which shows that after antibiotic treatment, the uptake of vaccination is more sluggish or not as high as in normal state. I'm pretty sure. I've never looked for this, but I can even predict, not only suspect, but even predict that such study or studies exist, because it makes a lot of sense that it's an interaction how your immune system is influenced by the microbiota, and this balance is then destroyed or skewed by the antibiotic, and then the uptake of the vaccination will be different and not beneficially different.
So it's interesting because with an mRNA vaccine, you're going to have essentially a systemic response, because this is going to get produced pretty much everywhere, as far as I understand.
Yeah, but that's probably a good thing.
Right. So you're going to get a systemic response, and it's to the spike protein, right? They're using it.
The potential benefit of the mRNA type of vaccines, is that it will also stimulate the respiratory mucosal immune system, which regular jabs, traditional jabs, don't do.
Okay.
So I think there is a chance that actually the mRNA vaccines will be more or closer to being sterilizing than traditional vaccines.
Okay.
There are also potential issues with the mRNA vaccines, but actually the efficiency may be really higher than traditional.
Yeah,
types
and
of
what do you
vaccines.
make of that? Because I have to say, it on the one hand, it is impressive when you have a 90% relative risk reduction in viral transmission between the groups. But when I look at the endpoint of viral transmission, instead of ICU or hospitals admissions, I have to say I'm much less impressed, and I'm not sure if it's convincing enough to be given.
Yeah, I think that probably people are so scared that they don't mind um, causing problems for others or even for themselves a little bit later down the road. Um, Blocking transmission should be the ultimate goal in a vaccination for respiratory viruses, because if you don't block transmission, you put a huge selective pressure on the virus. So you keep the virus circulating, and if you use a mRNA vaccine, for example, but also the adenovirus vector uses only the spike protein. So from this perspective, it's exactly the same. You use a very narrow range of antigens, and meaning that you put a huge evolutionary pressure on that part of the virus, or on those antigens, while at the same time those parts are already the most mutagenic. So those are reported to be undergo the highest rate of mutations already
Right.
without the pressure. So if you put even higher pressure on this one, that's potentially not a nice long-term solution. But the
Okay.
producers or even health professionals do not even claim that these are long-term solutions. So I
Right.
think
That's true.
these vaccines were rushed to market to prevent or to substantially reduce the death rate from this virus. That was the initial goal, and I think there are other vaccines in development already. But I see some chance that these mRNA vaccines can substantially reduce transmission
Hmm.
as
Yeah,
well.
I will consider it a huge win if it has no serious side effects. That
Yeah,
will be my main concern.
side effects we will see in 2023. That's when the trial ends.
Right.
Unless somebody terminates it early, for which I see a very high chance now,
Yeah, I
because
was about to say.
they will deem the placebo group not getting this life-saving
Right.
virus
Of
unethical,
course.
and
Where have we
then
seen
they will
that
convert.
before?
They will, yeah, they will convert everybody to the vaccine group, and then you lose basically you lose your control group for long-term
Hmm.
follow-up, and then we just won't see anything about long-term results just through the vaccine problem reporting system, which is rather flawed in
Hmm.
itself. So that then it would be difficult.
I have
I'm
to
wondering
say,
what
did
these people
you? Yeah.
signed actually in the placebo group. What kind
Right.
of agreement? Because if they agreed to be kept in the placebo group until 2023, so in the long term,
Uh-huh.
then there is not much to do. They should keep their agreement. They
Right.
should keep their agreement, but I have no clue.
But we already saw, antibody-dependent enhancement in two different animal models, actually, with the coronavirus vaccines. So I'm not totally reassured on that end yet.
That's I think that's mainly a problem with non-neutralizing antibodies,
Yeah.
and the benefit of using such a narrow range of antigens could be, which is very specific to this virus, could be exactly that it will produce low numbers of non-neutralizing and high numbers of neutralizing antibodies.
Mm.
So if it's neutralizing, then it binds to a region of the virus, the spike, obviously, because only the spike is administered in
Hmm.
these vaccines. So it binds to the spike in a way that it prevents the virus from functioning, from merging with any host cells. And
Hmm.
in that case, I don't see huge potential for antibody dependent enhancement. Because the spike is the most of the spike is really specific to this virus.
Okay.
So, but
let's hope you're right.
there are other areas, other antigens on this virus or in this virus which could be exploited the same way. So I think in the long term, the safest way would be to include as many very specific antigens as possible in a vaccine, and then we kind of distribute the risk over many antigens.
Okay.
Not only we already there were just recently read a preprint which reported a single amino acid mutations on the receptor binding domain, which is the outer facing part of the spike protein, which made both of the antibodies in the what is it called, it begins with an R, the drug company who has this antibody mix. Forgot the name.
Oh yeah, well anyways.
It's similar to the cosmetic company Revlon, but I don't remember. okay, doesn't matter. But they have an antibody cocktail with antibody cocktail which Trump also got. This antibody cocktail, and it consists of 2 antibodies, and a single amino acid mutation renders both of these antibodies becoming non-neutralizing.
Ouch. That's pretty specific.
That's a single amino acid mutation already out in the wild.
Well, at least there'll be a lot of happy drug companies to produce more antibody. Well, I would say, yeah, antibody test companies to produce much more kits, much more tests, always changing. It's going to keep them busy, I guess.
Yeah, I think they have a chance for at least another round of vaccines, for a second upgraded or improved or whatever. Perhaps they will make it for a third one. Who knows? It's a good business model anyway. So I believe in my own immune system. So I'm not lining up for any vaccines at this point.
Yeah, me neither.
I will think of my wife. She's most likely through the wild infection, but nobody was cooperative to prescribe an antibody test among her doctors, so we cannot know for sure. I will think about it, but
yeah, it's not a straightforward decision, I would say, at this point, just to put it mildly. But yeah, I just hope we can, end this lockdown. If the vaccine is a bad excuse for it, so be it. I don't really know what to say. You take what you can get, but the number of unvaccinated kids from tuberculosis and cancers and diabetes, heart attacks, and all that's coming up through the health system, I think it's a scandal, and we have to stop that wave. So I don't know what role the vaccine will play. We'll see. I'm not lining up to get it. You aren't either. I have nothing against.
But those people still scared. Those people who are still scared, let them do it. I mean, it's their choice. Sure. So I'm not.
I'm more worried about the people who are not going to be choosing, like the kids or the elderly. That's, I think, where my concern lies mainly, because they have no choice in the matter, unfortunately. I don't know. It's I just hope we don't have the health passport and all of that. That's
you can never know. It's in the works, I think.
That's really scary. Much scarier than the vaccine, even, I have to say. To be controlled about where you go and all of that.
I'm gonna buy a farm with my own cattle and then goats and then sheep and whatever, and then I just stop caring about traveling, and I will dig out a lake and I will bath in my own lake and don't care about travel.
Exactly. Have your own probiotic from the soil and the animals.
I'm already fermenting my own
Yeah.
kefir, so it's
Oh,
just
nice.
step to
Nice. I hope
have my
to.
own milk.
Yeah.
Have my own milk, and then we are one step further into being independent of these criminals.
Right. Yeah, I thought, you know what? The only intervention they would have had to have done during this whole Corona episode is just stop subsidizing the junk food industry, and in a couple, maybe probably 1 month, you'd have a massive fix to the excess deaths. Have everything plummet.
We've had 9 months already, so
Yeah,
in nine
you had
months
yeah,
you can lose 100 pounds, 50 kilos easily.
yeah,
Some
absolutely.
time back I lost 70 pounds, 30 something kilos in 5 months.
That's
So
massive.
it's possible, absolutely, if you are determined, and if you are really scared of this virus, you should have done it already.
Right. Yeah.
If you have that excess weight, why not?
If yeah, if the fear. Yeah.
Just yesterday I saw on local TV that famous, yeah, to some extent famous musician, um, and yeah, skin of color, they are more susceptible for weight gain and these kind of things, and to metabolic diseases. And the sad news was reported that he died of COVID. And you see the pictures of the guy put up, and then they say he had no known comorbidities.
Right.
The guy was like 170, like 6 foot tall, and 150 kilos, 330 pounds.
Oh wow.
He had no known
Known
comorbidities, and you see the
comorbidities.
pictures. You see
Wow.
the photos on TV, and you start thinking, okay. And then you hear about some friends working in ICUs that 90% of the people put on ventilators are overweight and obese.
Mm-hmm.
And this is the first you recognize that those people on ventilators are 90% overweight and obese.
Yeah.
So this I think was already clear in March. And if you haven't done anything in 9 months, there's nobody else to blame. It's not to blame anybody who is infecting others, or young people going out and
Mm-hmm. Living a
partying
life.
or whatever. This is always what young people do. What is not always the case, or it hasn't been always the case, that 88% of a population is metabolically sick. The
Yeah.
adult population, that's the US, in 2019, 88%, and 60% of kids are metabolically unhealthy.
And I would say
That's
that's on the low end estimates. I honestly, because if look at the people who are having a DEXA scan for fun and telling us about it, they say that the machines, the guys have never scanned someone without significant amounts of visceral fat. Like that they're the first people who barely don't have any visceral fat show up on the DEXA. I mean, people like they don't even know what normal looks like anymore. Like we are so far from that I think, yeah, your estimate of 88 is true, and probably even more jarring than that. It's probably even worse.
yeah, we
Completely.
like to call. I think some research team coined it way back several months ago, that it's a collision of two epidemics.
Yeah.
It's the obesity epidemic and the viral epidemic.
Mm-hmm.
And we could add, perhaps it's the collision of three epidemics. It's malnutrition and vitamin D deficiency,
Yeah.
then obesity and metabolic disease, and the virus. So
And
when you have these
isolation,
three,
yeah, you add isolation on top, it's the cherry, because isolation depresses the immune system. I mean, it's a basic need that's not met. It's a depressant in all senses of the word. um, it's terrible for you.
yeah, it's no wonder that previously we had very severe. I mean, if you think of influenza epidemics of '68, Hong Kong flu, and whatever flu there were, 50 something, there were huge epidemics, influenza epidemics, and a million people died or more, in half the population in one season. So it was more severe than this virus. And considering the much better health of the population, on
Right.
top of the lower number of the population, that was a crazy deadly
Yeah.
virus. And this
Yeah.
shouldn't be that deadly virus, but the high level of sick people in the population, and of course the increasing age on top. Because I just read a paper, I think yesterday, that between 2000 and 2018 probably, so in 18 years, the number of above 65-year-old people increased by 60%.
Oh, wow.
So it's such a short period of time. So it's more than 60% of above 65-year-olds around the world. And that's a huge increase. So no wonder that we are more susceptible as a society, as humanity right now,
Right.
because we are more and more older people living around. There is no wonder.
Yeah.
I mean, it's to be expected. It will continue. If a similar virus, new deadly influenza strain appears in a couple of years, maybe in 20 years, there will be again 40% more elderly.
I think you're
So
right. We're not
it
coming
will
to
happen.
terms with the demographics. We're not coming to terms with the health system we have and how we failed at people. So when you put both together, you have a failing health system, meaning the more technology it has, the worse the ratio of the outcomes that it can provide as the technology gets better. And at the same time, you have an aging population. And I think the collision is a good picture for it. Because what like you said, it's going to repeat. It's going to repeat with this coronavirus or something else, and we're
Yes,
going to see it. And maybe
especially.
the IFR of 0.27 could be 0.15 if we, did something better on that end. I mean, it's a big difference in relative terms, So
Especially if lab safety remains this poor, it will happen again.
yeah.
Did you know that SARS, the first virus, after the pandemic, after it has been contained or disappeared or whatever happened, was released from labs four different times?
Really? Oh my God.
So between 2004 and 8 or something like that, SARS, the first coronavirus, which was much more deadly, it had a CFR of close to 10%. It was released from different labs, at least what we know of.
Right.
These we know of. It was released from different labs 4 different times.
It's not funny, but it makes me laugh because it's so ridiculous. It's like what we're doing with the atomic, with the nuclear weapons, where a couple of times, if it wasn't for the common sense of one commander, then we would have ended up blowing ourselves up already. Yeah. So it's I don't know what it's going to take, but I just hope we can get out of the lockdown and start focusing on removing those excess deaths of misery and despair in unvaccinated children. I think
Yeah,
that's
funny
a
thing, you're not the first person I talk to who is really who really expects a quick spread of vaccinations just because of removing the interventions,
right.
the lockdowns and similar masking. But some people, health officials, already started talking about long-term masking because
Yeah.
it will solve all problems of humanity, obviously.
Yeah, the problem is just to be consistent on the matter, you're going to have to sound like an anti-vaxxer, an anti-masker, because you have to be able to point out when things don't work or they don't work in the way that's being presented. And there's no way not to sound extreme in this climate. So I think it's just, we've seen this in nutrition already, so it's not new for you and me. It's probably
We
new
are
for a
we
lot
are
of people.
we are pro-vaxxers right now because we expect that it
Right.
will really remove some of the
Exactly.
pressure.
Very much so. We will sing their praises far and wide. Um, yeah. So we'll see what comes of it. But I'm looking at who's making what predictions, and I'm keeping track. And so far, I have to say I'm very impressed by what Ivor is doing with Dr. Claire Craig and Joel Smalley, and some others out there who are doing some good investigative work using some publicly available statistics and doing us all a favor and exposing a lot of the nonsense. And Kevin McKernan, who's been excellent on the PCR and other testing technologies. So there's there's good people to
Yeah,
follow out there. So
I
make sure
think
you
it's
follow. Yeah.
I think it's always useful to keep an open mind, though, because I see that some people fall into a position
Right.
early, which is not fully substantiated, and then they are very reluctant to move away from that position if the evidence changes. And I think there have been some changes to the evidence base along the time this year. So, for example, we discussed the herd immunity threshold,
Oh,
and
right.
some
Yeah.
people put out there these 10-15% levels, which even I found a little bit too low, to be honest. And it seems that it's indeed closer to the 40% mark, even with the heterogeneous spreading factored in. So I think we must be open and always skeptic. Skeptic not only with in connection with what pushers say, but also in connection with what our friends say. It's
Right.
important that okay, let's step back one little step and think about it
Right.
before just jumping on the bandwagon.
Yeah. Yeah. It's much easier to jump on the bandwagon after you don't have to run next to the train. So it's I think people take the
Sure.
easy way out. Yeah. All right.
Yeah, skepticism is always nice, but skepticism should be all around. So it's not specific to one idea, but be
Right.
skeptic about almost everything.
All right. Indiscriminate in that sense. All right. I will pause the recording here.